Yes. Good evening, participants. Am I audible to you? So, welcome to the fifth session of internship and certificate course of artificial intelligence and machine learning approaches of drug and vaccine design in cancer. So, today we'll be discussing about the chapter 5 about molecular docking. Okay. So, am I audible to you? Yes or no? Any interact? Am I audible to you, participants?
Yes, sir.
Then we can recap the last session contents. So can we discuss about the last session topics? So just type the answers in the chat box. What exactly did we discuss in the last session? So last session topics, molecular talking just started, very good. But what exactly we discussed in more? So we have discussed various methods of computational drug design. So we have started something about molecular docking. Then we discussed about molecular dynamic simulation. Then what else did we discuss? What hands-on experience we have seen? Very good. We have also seen Swiss ADME. Once again, we recap, isn't it? So we have seen various drug databases. So what are the various drug databases we have seen? We have seen Zinc 15, we have seen Drug Bank, CHEMBL, and also we have discussed about therapeutic target database, isn't it? So that's what Karthik has replied. Therapeutic target database, TTD. And what else? What are the other softwares we have seen? Online tools we have seen? Any other online tools? In Swiss, we have seen so many tools, isn't it? Other than Swiss ADME, we also seen Swiss target prediction. Good. Only one answer I got. What else did we discuss in the Swiss other than ADME and target prediction? Did we see any docking tool in the Swiss? Very good. The answer again I got from the Sania. So the answer is Swiss dock. Okay. So here you have to simply input two files. One is ligand and the other is the receptor in 3D structure. Okay, so both files can be of PDB format. So if you input the files, obviously we can find a kind of an output after the doc process. And any other tools missed out? Good. I think when you're following up, so I'm glad to take you to the fifth session more in detail about molecular docking. So what is molecular docking? It is the computational procedure to predict the conformation of a protein ligand complex. So generally we try to check the binding affinity of a ligand. The ligand is generally a drug inhibitor. So we try to bind with a macromolecule. The ligand is the micromolecule and macromolecule is the protein. And if you see the molecular docking protocol, we have two components, search algorithm and search function. Generally, what is docking? I told you we have a kind of a target. We try to fit a kind of a ligand and check how well it has bound with the receptor or not. Okay, so simply in terms of 3D structure, if you see this kind of a protein is in surface format of a protein, and here you can see it is in the form of wireframe model. Okay, so this is in the form of a wireframe model. That is, a ligand is in the wireframe model. We try to fit this ligand wireframe in this surface of a protein. So you can see it's binding only in the hydrophilic surfaces. If the ligand is buried inside, you might not have seen the ligand. Okay, just because we are seeing the ligand outside, it is a hydrophilic binding site. So if you see the docking, we can see various types of docking. One such docking is protein ligand docking. So in Anna University, many scholars, even when I was studying, when I studied 20 years before, we are practiced UCSF dock. UCSF dock in HA graphics operating system. There is one operating system called HCA graphics. In that operating system, we have used USEF dock. Okay, there is a free version, and there is also one more dock called Gram from Sunny company, and ICM dock from also. There are plenty of tools, thousands of tools are there for docking ligand with the protein. And if you see the protein docking, few softwares like 3D dock suit, bigger plus pro dot hrng head of hex. These are meant for docking protein versus protein. Here, whatever I mean protein, one is a micromolecule we consider as a micromolecule, and the other, which is a large, we consider as a macromolecule. So in this case, the ligand is also a protein. And also, the docking you can find in various biomolecules. If the DNA is docked with protein, DNA, in DNA binding proteins, you can see DNA is docked with the protein. And in RNA binding proteins, RNA is docked with protein, isn't it? So the DNA and RNA are micromolecules considered with protein as a macromolecule. Okay, so there are so many biomolecules dock with bind with each other. So then we have to partially differentiate what is micromolecule and what is a macromolecule. And if you see the different docking methods, I initially told you there are some freewares like UCSF dock, Autodock, okay, and Affinity, Flex. These are all, of course, commercial ones. But if you want to download these kind of softwares, the homepage is mentioned in this. Okay, the URL of the homepage. So the UCSF has to be downloaded from doc.com by your tools from UCSF, and from Autodoc can be downloaded from Scripps Educational Institute. Okay, scripps.edu. And Affinity from Acer is Flex from Biosolvit, Gold, ccdc.cam, and Fred from isopen.com, Glide. This is a Schrodinger software. In a Schrodinger package, you have one docking software called Glide. This is used for only docking, ligand receptor docking. So docking and dock we already discussed. Prediction of the binding energy of the ligand receptor complex. How the binding energy of the ligand binding with the receptor in the complex is determined. So the basic equation is thermodynamics. Okay, we determine through the change in Gibbs free energy. The change in Gibbs free energy is defined as the difference of change in enthalpy to the product of temperature and change in entropy. So del G equals del H minus T del S, where if you see this del G can also be calculated for the final change in Gibbs free energy can also be calculated from the initial change in Gibbs free energy minus the RT ln KD. R stands for universal gas constant, T for absolute temperature, KD for dissociation constant. Okay, so this is how we calculate the binding energy through the Gibbs free energy. If you see the molecular docking is represented in various areas of biology or biotechnology. If you see in immunology, so in immunology we can find these kind of interaction, isn't it? Many we check with antigen antibody interactions. If you see the interleukins, some examples for interleukins, IL-2. So can I ask you a few questions? Because even one should have a knowledge on immunology, pharmacology, etc. for this course. So kindly provide me examples for interleukins. Or if you know any examples for antigen antibody interaction, you can type it. Do you know any example for antigen antibody interactions? Any example? An example of antigen and example for antibody? IL-4, very good. In IL-4 is interleukin 4. We have IL-4 ligand, okay, binding with the IL-4 receptor. Okay, we have this, and even IL-12, I think we have IL-4 ligand binding with the IL-4 receptor. So there are so many kind of cytokines, especially interleukins, where you can able to find a ligand and a corresponding receptor. Okay, so in that case, you can find if a receptor is considered as if a receptor is considered as antibody, then antigen is your ligand. Okay, then if you see in enzymology, okay, a substrate binding the enzyme. Okay, and if you see signal transduction, cell signaling areas, okay, can able to see a molecule is binding with the transport molecule. So, okay, this is a macromolecule, this is a micromolecule. Do you have Have any idea about an example for this? For this, you can take an example like hemoglobin or myoglobin. You know, the hemoglobin protein, which is a macromolecule, can carry oxygen. You know, in hemoglobin, we have iron molecule in the form of a complex, okay, which can also carry oxygen molecule. If hemoglobin is carrying the oxygen molecule, then we call that protein as oxyhemoglobin. If hemoglobin is carrying carbon dioxide, we call it as a carboxyhemoglobin. Okay, so the transport molecule is your transport protein hemoglobin, whereas the molecule binding with it is the gaseous molecules like oxygen and carbon dioxide. That's an example. And similarly, in cell signaling or general signal transduction, even whatever I explained you, like you can better take the acetylcholine binding with the neural receptors. If acetylcholine bind, obviously we find signaling, isn't it? Yeah, how the brain triggers. The brain can trigger if acetylcholine binds with it. But alternately, if dopamine binds with the receptor, you find a kind of analgesic kind of effect, isn't it? So all these things, okay, the signaling or whatever we study in various areas of biology, okay, so that we call it as a molecular recognition. But whereas in computationally, we call this as molecular talk. Understanding molecular recognition. If you understand, if you see this recognition, we can, in pharma, we call the terms, we can call the ligands as inhibitors and sometimes as antagonists. Okay, but if it emulates the recognition, if it blocks the recognition, we use these terms. But if it increases the recognition, increases the signaling, then obviously we call it as an agonist or a recombinant protein. Okay, so there are different technical terms based on blocking or increasing the signaling effect. And if you see in docking, we simply can able to check whether how effectively it binds. But the increasing or emulating and blocking can be studied only through in vitro or in vivo studies. Okay, so only through that, only through the microarray, then we can studies we can find the expression patterns. And if you see this molecular docking methods, initially the theory was laid by Emil Fischer. He has developed lock and key model, and so the scientists, the computational scientists, those who want to develop a kind of a software for molecular docking, they have used these kind of principles, 1890 lock and key model. And Koshland has used induced fit, isn't it? He has come up with induced fit. So later, the software developers came up with these kind of models for molecular docking. Later, in 2003, many scientists are focusing on to develop molecular docking tools using conformational ensemble. The author, the father for this conformational ensemble is Boyong Ma. If you see lock and key, see if you consider this as a lock, a low color, which is a receptor, a macromolecule, the key is your ligand. Okay, if only if the conformation is exact, then only a lock can bind with the key. Okay, this is what Fisher key and lock model. But in this fit is like just opposite. So even if you have a ligand in white color, like a ligand white color and blue color, if you fit exactly, it won't fit because you need a kind of a conformation change. The conformation is obviously changed when uh, if both the receptor as well as ligand is considered as flexible. Okay, here in this case, the ligand and receptor are rigid. But in induced fit theory, we consider both ligand as well as the receptor are flexible. Then, if it is flexible, you can able to fit the ligand with the receptor. That is induced fit theory. There is one more model called conformational ensemble model, where you are using, of course, an induced fit model, but here you try to talk with multiple conformers of a ligand with a receptor. What you do is you can keep a protein as a flexible or rigid, but the ligand, okay, you generally keep it as a flexible, okay, and you try to dock with the receptor if in various conformation, you try to dock a ligand with the receptor in various poses, and the pose where you can find the negative maximum energy. is what we consider as global energy. So in this picture, okay, you can able to see there are totally 5 conformations generated, and there is one some n conformer. So if you see which is the best uh, ligand docking with the receptor, this is the answer. Okay, the 2. So this is the negative maximum when compared to any other conformation. So this is the best fit. Okay. So this is how we can able to describe the docking efficacy or efficiency through the conformation and ensemble methods. And if you see the kind of arrangements, so we generally here in 1890, I told you in lock and key it's a rigid one, but whereas a slight modification um, is incorporated in induced fit, but a large rearrangement. A large rearrangement, I mean, a ligand is just having a kind of a flexible kind of interactors. So whatever functional groups attached with the ligand is more flexible. It fits in such a way with different poses of in a receptor, and so you get so many kind of uh, binding energies. Okay, so only through the global energy you finalize it. And if you see the docking, the types of docking, okay, the docking, as I told, the ligand is not a must that you should have a small molecule. You can also have a macromolecules like DNA and RNA. And if you bind DNA with protein, DNA binding protein, RNA with protein, RNA binding protein. You can also dock protein with the protein, isn't it? So that is protein-protein interaction. And if you see the molecular complementary in computational docking, we have a shape complementarity. So here you can see exactly like in a puzzle how one piece is fitting with another piece. But here, if you see the chemical complementarity, you find the polar group attracts polar, non-polar attracts non-polar. I think you know this solvency principles. Okay, so we follow all this chemical complementarity in the solvency principles. But also we find a positively charged ion generally attracts with a negative, isn't it? And negatively charged with positive. So even charges are taken into account in chemical complementarity. So what kind of interactions you can able to observe? So not only this polar and charges, but also based on the hydrogen bond sharing. If you have a aliphatic hydrogen bond acceptor sharing with hydrogen bond donor, only one hydrogen bond is shared. But if you take an aromatic of 6 membered ring, 6 hydrogen bonds are shared. If you take a hydrophobic, you can find this kind of interaction. So, which is comparatively better with the electrostatic. And if you see the energy, I told you there are different, we can get different poses in the conformational ensemble model. If you see, if this is your ligand, okay, this ligand is not best fit with the receptor, but here in this case, the ligand is tightly bound with the receptor. So only we got negative maximum. But see here, in other poses, the ligand is not exactly binding with the receptor. So you are getting some energy, but it's not negative maximum. Okay, this is how we screen the best conformer of a ligand through docking process. And if you see the types of docking, the docking can also be classified based on the rigidity and flexibility of the ligand and protein receptor. So if you see protein as rigid and ligand as rigid, the docking is possible. So we can also see the flexible ligand is docking with the rigid protein. But one thing which is not possible is keeping a ligand rigid and making the protein as flexible. Only this docking alone you can't find in any softwares. But nowadays, the commercial softwares are using this kind of a principle, which is more complex, that is keeping your ligand as flexible and protein receptor as flexible. So most of the commercial softwares use this complex kind of calculations to check the binding energy. But freewares generally we can see this kind of flexible rigid, something like that. Okay, even Autodoc works like this. Scoring methods. Even we have types of Autodoc, so most of Autodoc tools work like that. And if you see the scoring methods, the scoring like the binding affinity can be measured only through the score, the doc score. Okay, if you see Discovery Studio ligand fit, we have 8 different scores. We have 8 different scores, like we have PLP1, piecewise linear potential 1, and similarly PLP2, chain score, MMF score, torque score, binding energy score. We have so many kind of scores. Nearly totally, we have 8 kind of scores which are generated from Discovery Studio ligand score module. Discovery Studio ligand fit is one of the module to dock a ligand with the receptor, but ligand score is used to score the binding affinity of the ligand and receptor. One example I mentioned. Okay, so if you see this kind of a scoring methods in Autodock, which I am going to show you shortly after completing this theory session. So we have only internal binding energy score. In Autodock, we have only one score, that is internal binding energy score. But if you see in tools, commercial softwares, we have plenty of scores. Then what are these scores? Why should have a plenty of doc scores? If you ask if you ask me, I can tell you that the scores are calculated in combinatorial fashion with different physical and chemical properties of ligand and the ligand receptor complexes. You see, number one here, whatever factors I shown, number one is interaction energies between the two molecules. This is one important factor. And number two is the desolvation and solvation energies associated with the interacting molecules. This is a parameter two. And third is entropic factors upon binding. That is, a ligand receptor entropic factor is calculated like this. Lipophilic index, and then polarity, and then pKa value of a ligand. Likewise, we have so many physicochemical parameters, isn't it? So all these parameters are taken in consideration, and maybe if PLP1 take care of lipophilicity and lipophilicity and solvation, and may the doc score is taking they might have calculated with bonding interactions and non-bonding interactions. So it depends. Okay, it depends like what parameters they have chosen to calculate what type of scores. Okay, so it's up to the software developer. And if you see one such recently published article, that is, if you see in Letters in Drug Design and Discovery, an Infrastructure Journal from Bentham Science Publishers, I just talk a plant compound called Vithaferin A with two important receptors for treatment of cardiovascular diseases. So, you know, actually this Vithaferin has shown a negative maximum energy with these two receptors. So not only used molecular docking, but they also used simulation to finalize, to validate the kind of a binding affinity results with respect to time. So one such extracts of 3D images of how with a frenzy is docking with a protein receptor. So whatever you find a ligand, the ligand is in the wireframe model. If you exactly see, this is in the stick model. The ligand is in stick model, and the protein binding amino acids are in wireframe model. So similarly, in 2023, a docking article you can find an information on docking a plant compound with two important marker receptors of hepatocellular carcinoma. So the docking energy as well as the simulation results are mentioned in this article. So these are the case studies. In that article, you can see this kind of a 2D interaction of Virinon B docking with the epidermal growth factor receptor. Okay, and this is a kind of a apoptosis, kind of a marker protein caspase 3. So just dock with it, and this kind of a 2D interaction gives information on that. And now I think I can take you to the hands-on experience in Autodock protocol. So if you see the Autodock, okay, so we have steps. First step is preparing the protein file, and major step second two is about the preparing the ligand, and third step is to prepare the flexible residue file. Fourth step is running autogrid calculation, and fifth step is preparing the docking parameter file. Okay, so these are major steps, and finally analyzing the docking results. Okay, so after this we'll be seeing how to analyze the docking results. So nicotinic receptor, muscarinic receptor, yes, so many examples are there, isn't it? So to know more about kind of protein-protein interactions, we can also see textbooks like pharmacology, different pharmacology textbooks, Ken Willy, Pinfords, etc. Okay, now I take you to the tools. If you want to work on Autodoc, okay, first I can show you a home page for Autodoc. So Autodoc suite can be downloaded from autodoc.scripts.edu. So this should can be the software package can be downloaded from. And if you see the Autodoc, okay, we have types of Autodoc. Number one is Autodoc 4. Number two, Autodoc Vina. Number three, covalent Autodoc. Number four, Autodoc PYRX. So like this, we have so many products. Okay, so Autodoc for downloading this, you can download Autodoc 4 or Autodoc GPU. Then how about Pyrex and other kind of versions? You have to go to the specific website and you can download these kind of tools. Autodoc Vina can be downloaded from vina.scripts.edu. Even Autodoc Pyrex can be downloaded in the corresponding website. PYRX. So this can be downloaded from pyrx.sourceforge.io. Okay. So we have different Autodoc tools in different websites. So you have to. So if you see all the Autodoc tools or commercial thing, even whatever they mention Autodoc Pyrex as a free, okay. After 15 days or 30 days trial, I think 15 days trial, obviously it asks you to renew with the license, but we can skip the license and we can still work with the kind of Autodoc. But I think one or two Autodoc you have to strictly pay unless you can't skip and work on it. Okay, so this is something about Autodoc tools. Okay, and if you see this kind of Autodoc, for installation of Autodoc, one should install, okay, EDU PyMol, Python molecular viewer. And I told you you can download Autodoc suite from the home page, isn't it? So there you have to download MGL tools. MGL tools. Both Windows 32, we have only Windows 32 kind of exe file, but even it works in 64-bit version of Windows operating system. So to work Autodoc in Windows, you have to download EDU PyMol in the PyMol website and MGL tools in Autodoc website, and also Autodoc suite. There is also one more suite software exe we can download from that Autodoc suite. And finally, Chimera. Chimera can download from the Chimera website. Okay, so Chimera tool. So totally, you have to download and install 4 important exe files. So I can type what are the most important: edu pymol, Autodoc suite. I told you download and install Chimera. After Chimera, what is that? So MGL tools, MGL tools. So if install all these 4 softwares, it can work well with the. Sir, can you tell these laptop specifications required to download? Actually, to run the Autodoc or Autodoc Pyrex, I suggest you to use i5 or i7, but even it works with i3. Okay, so i3 is better. Could you provide links to the above files? Yes, it's downloadable actually. So whatever I've shown, if you want to download, if you see the Pyrex also available in other operating systems, Linux, okay, it's also available in the Macintosh. If you are using an Apple laptops, if you want to download the kind of a free version, just click here, click download. So instead of relying on some other exe files in non-trustable websites, use this kind of a URLs. Sourceforge dot. So some of the URLs, okay. Likewise, initially I showed you Autodoc, isn't it? Autodoc 4. So Autodoc. So you can click the downloads, Autodoc 4. So you can download the files from this website. Here you can have Windows. Okay, so this is one more URL. Okay, so likewise, you can go for Chimera. So you can also download Chimera like this. So in trustable websites, you can download. Okay, so I provide some links. You can download similarly Chimera and you can work on it. After downloading an installation, so what are the icons? What are the icons you can find in your desktop? If you see if you after the installation of Autodoc, okay, you can see this tool. This is Autodoc tool. A ligand finding is binding with the receptor, the blue color in blue color. So this is after installation of Autodoc suite, you find this kind of an icon, and you also find CADD icon, and you can find this Vision 1.5.6, Vision 1.5.6, and Python molecular viewer, PMV, Python molecular viewer. So totally, you can able to see these four icons: the blue color, this orange color, this kind of a Python molecular viewer, and Vision. If you have these four icons, and even these softwares are installed in random. So you can install first Chimera, and then you can install Pymol, or even you can install Autodoc, then you can install Pymol. So however you want to install, you can install it. Okay, there won't be any problem in downloading and installation. Okay, so if you have installed all this, okay, I suggest you to how to go for doing a simple docking process. I can supply you with this kind of a tutorial, Autodoc 4 tutorial. So I send it to coordinator, Professor Praveen sir. He'll be sending it to you. And also tomorrow, I'll be sending you the different tools, what we are used from the beginning, and also what we are going to see later. So I'll be giving you one more document. Okay, so go through that kind of an examples and work on it. So now a tutorial I developed a kind. There is a tutorial for Autodoc. Okay, so we have to follow this tutorial for doing the docking process. Unlike SwissDoc, just providing a ligand and receptor as input files, click submit, you get a result. But here, you won't get with the simple steps. We have to follow these steps. Each and every major step has some sub steps, and we have to stringently follow the procedure. If you alter any part of your procedure, you won't get a result. If you forget to do one thing, one sub step, obviously you won't get an answer. You don't know unless you work for a minimum 5 to 10 minutes on this procedure. Okay, then you have to restart from the beginning and you have to work after diagnosing or troubleshooting where you made wrong. So okay, now let me start with Autodoc. Okay, some precautions we have to take. Okay, so we have to stringently follow the protocol to get a result in Autodoc. That's what I want to mean. So here initially what I did is after installation of Autodoc software, I kept one demo participants. Sumanta. Okay, so let me just continue my session. So I was explaining about how to dock with Autodoc. So I was explaining about the steps, and initially explaining about the softwares to install the Autodoc, isn't it? Now I take you to the file preparation. So initially, if you want to prepare the files, okay. So if you have installed Autodoc in the program files, in the Autodoc 2 folder, you can find these exe files, Autodoc 4.exe, AutoGrid 4.exe. These two exe files, you better copy and paste it in one demo folder file. I just in E drive, I had something like demo 10, where I pasted these two exe files. These two exe files, again, I'm telling, I got only from the program files of the Autodoc after installation. Okay. Then I'll be taking one ligand. The ligand I have taken is Inginavir, and I have taken a receptor HIV protease. You know how I got this protein and ligand? So this protein and ligand I got from PDB website in a single PDB file. So if you see this protease, okay, I just downloaded the ligand receptor complex, just split the protein separately and I have splitted the ligand separately. And now I'm like to dock the ligand with the receptor through Autodock. So now we can see that in a demo folder, I have 5 files. One is autodoc4.exe, the other is autogrid4.exe, one is ligand.pdb, and another is receptor.pdb. The ligand is indinavir and the receptor is HIV protease. This is an example, isn't it? I'm just showing with an example. And I have also a Autodoc4 tutorial. Okay, now I start explaining you with the Autodoc 4 software. So Autodoc 4, if you want to start, okay, simply double click this icon, the blue color icon. If you click this, okay, you can find an Autodoc is getting open. So it's getting loaded. After getting loaded, click remind me later because I don't have any licensed version. So after clicking this, you can find a Autodoc window is open now. Okay, to work on it, okay, so we have to take steps one by one. The first initial step what we have to make is file preferences set. File preferences set. In this, we have to set the path for all the files where we had in the repository. You remember, I told you, we kept these files in the E drive. The demo 10 folder is kept in the E drive. Why I kept in a folder? Because while doing a docking, you get so many intermediate files. Whatever intermediate files you get, including a docked ligand receptor complex, you can have in a single repository, in a single folder. So I kept in a single folder, and I'm going to feed like whatever intermediate files and output files are going to generate has to be generated only in this startup directory. So I just pasted this URL of the folder. Now click make default. Next, click set. Make default, click set, and click dismiss. So the first step what I made is preferences set. Okay. So if you see the tutorial I might have mentioned. So first on the file preferences set startup directory. What is the next step? Prepare the protein file. So what you have to do, you have to prepare the protein file. Okay, so how to prepare the protein file? File read molecule, select protein name. File read molecule, select what is the protein name? Protease, isn't it? Click open. Now a protein molecule is shown in the window. Now what we have to go? So add hydrogens and charges. How to add? Edit hydrogen, add all hydrogens. So check for edit. Did you find edit? Hydrogens, add hydrogens. So default parameters has to be restored. No need to change any parameters. Click all hydrogens, no bond order, yes. Renumbering atoms to put new hydrogens, yes. Now click OK. So you can see a small change, a configuration change. The hydrogens are added in the protein in the visualizer of Autodoc. Then we go for edit, add Coleman charges. After adding hydrogen atoms, we have to add charges to it. So for this, you use the same edit, charges, add Coleman charges. You get this kind of a pop-up window. The charges are added. Click OK. Now we have completed up to this. The next step is to save the file as protein1.pdb. How to save this? File, save, write pdb. So how I told you, you have to save like protein1.pdb. So instead of protease, now I'm going to save like protein1.pdb. In E drive demo 10 folder, I saved it like this. Now click OK. So I have completed up to this. The first step is completed. What is the first step? Preparing the protein file. So the next step is preparing the ligand. For this ligand input, open ligand. Ligand input, open ligand. So I didn't find any ligand files in PDB-QT format. You know, in Autodoc, both the ligand and receptor should be in PDB-QT formats. If you have downloaded from AST or ASTF format, you have to convert as PDB. Then later you have to convert as PDB-QT. Okay, but initially we don't have PDB-QT files. So what I'm going to do is click all files. So the second step is to basically open the ligand. Where is our ligand? The ligand is here, indinavir. So this I'm going to open. Now a summary is displayed, some charges are added automatically and some aromatic carbons and rotable bonds are formed. Simply click OK. Now our ligand is added somewhere in the receptor. Now the next step is to go for ligand torsion tree detect route. Ligand torsion tree detect route. This step is completed. Next, ligand output save as PDBQT. Ligand output save as PDBQT. How they want to save as ligand1.pdbqt, isn't it? So I'm going to save exactly like that. Ligand1.pdbqt. So now this is saved in as PDBQT file. Only the ligand alone. Now we are prepared in the form of PDBQT format. So we have completed up to this. We are yet to convert our protein molecule as PDBQT. I told you both the input files, the ligand and receptor, should be in PDBQT format, isn't it? So to save the protein file as PDBQT format, we go for the third step. In the third step, we prepare the flexible residue file. Okay, so to understand, the macromolecule is made as flexible in this case. Okay, so a grid macromolecule. So grid macromolecule open. So here you have to choose only the macromolecule. But here I have ligand one. If the macromolecule is not shown, instead of PDBQT files, click all files. What is the recent file I have saved? The recent file name I have saved is protein 1. So click this and click open. This is the macromolecule, isn't it? The protein 1 is macromolecule. So I have selected, click open. Now you get a kind of a warning, some kind of a non-bonded atoms are there, some non-polar hydrogens are there. Simply ignore it by clicking OK. Now it's asked you to save a file. How we are going to save? Now I'm going to save protein 1 dot pdbqt. Okay, protein 1 dot pdbqt. So this is how I'm going to save my protein file at last. So I've completed up to this. Okay, I prepared ligand1.pdbqt. Now I've saved a protein 1.pdbqt. The next is to define active sites. To define the active sites, we take this step. Select from string. Select from string is here. Now, in the molecule list, you have to select protein 1. This is the latest protein. You have to select the latest protein file, that is protein 1. And in atom, if you want to select all the atoms in an amino acid, you simply has to type asterisk. And participants, ensure you click only once. The add, we have to click add, but we have to click only once. If unknowingly, if you have clicked add, and again second time if you try to click add, whatever steps you have taken from the beginning has to be repeated. Or else, if you proceed with the subsequent steps, you won't get a result. Okay, so ensure if you whether you clicked or not. If you have a dot, better close it and start from the beginning. Okay, now only I'm going to click add only once. Yes, after clicking add, you can see the conformation change in the protein. Okay, so after this, you click this place. That's it. So up to what step I have completed? I have completed up to this third step. The third step is fully completed. Now I'm going for the fourth step. Running autogrid calculation. You know why I have taken some indinavir HIV drug binding with HIV protease? The reason behind why I have taken this as sample is like this protein molecule is very small in size, and even the indinavir is a simple drug. So if I want to say, show you the results in a short span, I'm just taking this as an sample. But if you're working on cancer, if you're working on a particular protein, you download that protein and you can download the corresponding tag and you try to dock a single ligand with a single receptor in this Autodock. In this Autodock tool, you can dock only one single ligand with a one receptor alone. Multiple ligands docking with receptor is not possible in this software. Okay, let me continue with the fourth step. The fourth step is running auto grid calculation. So for this grid, set map types by choose ligand, select ligand. Even if I mention choose ligand, you defaultly use open ligand. That is easier for you for practice. So grid set map types by open ligand, select ligand. So how? Grid set map types by open ligand. So initially they want to open the ligand, isn't it? So you have ligand 1.pdbqt and protein 1.pdbqt. What has to be opened? The ligand. So ligand 1.pdbqt has to be open. Yes, we have completed this step. Next is grid box. Grid box. Okay. So for this, we have to click grid box. If you click the grid box, you can find a blue color window. Okay, if you just drag it, you can see blue, red, and green are there in X, Y, Z directions. If you click this, if you right click this window, the 40, the red color, you can find this uh, window is open. Here you can find the value as 40. But if you want to edit the values to increase the size of the grid, to enclose an entire protein molecule in a grid, then you have to click apply. If you click apply, all the things are inactive now. Now, whatever you want to change, you can change it. Now, I'm going to increase the size of the box in the X direction 150, 150. Yes, the size is increased. Now, simply click OK. The size of red color is increased. Now, I'm going to click green color. I right click the green. You get this window. Simply click apply. And you can provide some dimensions. I'm going to provide dimension maximum of 140, and even the value what I'm going to change is also 140. A large window, a large Y direction window is open. Finally, we have to increase the size of the blue color. Right click this, click apply. Now I just keep the value as 126. Let us see. Even if you have a large box, then it takes much time. You know why we are enclosing this protein molecule in the grid? The reason why we enclose a protein molecule in the grid is if you want to determine the lipophilic or hydrophilic binding sites, if only if you keep a protein molecule in a box, in a grid box, and I told you on yesterday, there is one algorithm called flood fill algorithm. If you pour water inside the box, wherever water is going to accumulate, then you can able to calculate the hydrophilic binding sites. You remember the Archimedes principle? So even the sign, what he did, if we in a water, if you want to determine the volume of a rock, he just dropped a rock, okay, in a glass of water, or a small rock in a glass of water. So how much water is displayed? He just measured that and he determined that's the volume of the rock. Similarly, here to determine the binding sites, we use a grid box to enclose a protein. Both the hydrophilic and hydrophobic binding sites are determined using this. But if you have a very large box, see here, the red is very large. So it takes much time for docking. So better you fit the protein to the desired dimension. Okay. I am checking whether if I reduce the value, whether it can fit or not. Still, whatever I used is a large value. Still, I am reducing further because if you want to run the Autodoc, it takes much time. So I'm reducing the size. Still, this is okay for me. And check how about the green? The green is very large. So now I close this and go to the green box and I reduce the size for green. So I click the value as 100 and maximum of 100. Yes, still, yeah, we are closed. Yes, how about red? Whether the red is large? If red is large, even I can reduce. Now I reduce this. Okay, red, blue, green. Now see, my entire protein molecule, the blue is not fit, coming out. So I have to increase the size of blue. I'm going to increase the size of blue as 135. Now it's perfectly fit. Okay, we have perfectly fit our protein molecule in this grid box. After doing this step, you have to click File, Close Saving Current. So File, Close Saving Current. I have completed up to this step. Now go for Grid Output Save File. Grid Output Save File as GPF. So how you have to save? you can save file as grid1.gbf. Okay, so I've saved this grid as grid1.grid parameter file, grid1.gbf. Next, we have to go for running the autogrid. So in Autodoc, in running the uh, doing the docking of ligand and receptor, this step you take much time. Run autogrid launch. Now I'm going to do that step. Run autogrid. If you click this, you get program path name. In program path name, you just have autogrid 4, but you have to have an entire path. So for this, you click browse. So you have to click autogrid 4, and if you click here, the entire path is shown. And also some kind of CMD command, you can find a path. The next is to select for parameter file name. So you have to click browse. What is the parameter file we have saved just now? grid1.gbf, isn't it? So default it is there. You simply click and click open. Now even you get like the URL for two different files. One is for log file, grid log file, other for the grid parameter file. And even you find an extension of URL in CMD. Now the next step is to click launch. If you click launch, you get a kind of a white window. This white window, if it immediately disappears, then you might have made mistake in any of the substeps. If white window immediately disappears, definitely that you made a protocol wrong. So what you have to do, close the entire window, start with a new folder, paste all the 5 files, a ligand file, a receptor file, autodoc4.exe, autogrid4.exe, and if possible, a tutorial. Okay, if you paste all these files and you can start from the beginning. See, my white window immediately disappeared. Not immediately disappear, it takes some time for disappearing. Now my molecule has completed the autogrid. You know, my configuration of laptop is i7 with 16 GB. So the performance is faster. So when I can able to complete within a minute. I think within 30 seconds we have completed. But if you're using i5, it takes maybe 1 to 1.5 minutes. And if you, the same files, if you're working with i3, it may take even 3 minutes and more than that. It's based on even your processing power of your laptop or desktop, whatever you use. Okay, now I have completed up to this step, 4th step. Now the 5th step is to run the Autodoc. So 4th step is to run the autogrid, and the 5th step is running the Autodoc. For this, you have to initially prepare the docking parameter file. So what is the substep? Docking macromolecule set rigid file. Docking macromolecule set rigid file name. So macromolecule file alone has to be get selected at present. So click protein1.pdbqt, click open. Now we have completed this step. Next, docking ligand, choose ligand. I told you wherever you find choose, click open. So docking ligand, open, select ligand. Docking ligand, open ligand. So what we have to open? Ligand1.pdbqt has to be opened. Click open. Now you get this kind of a window. Simply click accept. That's it. So I have completed this step. Next step is to go for docking search parameter genetic algorithm. You know, we are using genetic algorithm for docking a ligand with the receptor. So docking search parameters genetic algorithm. In the genetic algorithm, okay, you can also give your choice according to the poses. If you want the first best 2 poses, you can change instead of 10 runs, you can click 2. Like if you're going for a docking, defaultly it takes 10 runs. It means it shows 10 different binding energy scores. But the one which is negative maximum is there at the top. And number 2, the second negative maximum is at in the second pose. So generally, if you want to show the scores in the paper, you can take only one, isn't it? Not even the second score is needed at that time. So I simply restrict my docking runs as 2. Simply click accept. This is completed now. I have completed this step. The next is docking output American genetic algorithm. Earlier, what I did, genetic algorithm runs instead of 10, I have selected. I type 2 and click OK. Now, docking output. You are going to save this docking output in American genetic algorithm as doc1.dpf file. Now I am creating a doc1 as a docking parameter file. Click save. Earlier, how you created a grid parameter file. Now I am creating a docking parameter file to run the auto dock. Earlier, we run the auto grid. So the last step is to run the auto dock. For this, run auto dock. So instead of Autodoc 4, we have to get a URL for that. So click browse. We have already have doc4.exe. Click this program and click open. Now we got this and click here in browse. In the docking parameter file, select this doc1.dpf. So we also got a kind of a URL for it. And also we have docking log file name and even a URL is changed here. Now the step is to click launch. You click launch, you are running the Autodock. But this kind of a white window disappears quicker when compared to the running the auto grid. Running the Autodock takes less time when compared to running the auto grid. But again, I am telling you, the appearance and disappearance of the window is based on the size of protein molecule, the macromolecule, and the size of the ligand molecule you have chosen. Okay, but they should not disappear immediately. It takes some time to do the docking process virtually. Okay, and if the window disappears, then we can carry out on the other steps. Not yet disappeared. We are waiting for it. So meanwhile, I can show you. There is a sixth step, reading the docking log file. So in the fifth step, we have completed how to run the Autodock. In the sixth step, we see how to go for docking analysis. Okay. And after going for docking analysis, we can open the docked file in a visualizer. I have installed a BioVia Discovery Studio visualizer. So using that visualizer, we can see the ligand protein receptor interactions in 2D format as well as in 3D format. Now I completed up to fifth step, isn't it? Next, we proceed for the sixth step, reading the docking log file. How to make it? A sub-step is analyze dockings open. Analyze dockings open. So here you have to open doc1.dlg. Okay, so that's what they have mentioned. You have to choose the DLG. Doc1.dlg is open. So default, you get this kind of a window. Simply click OK. Now, if you get the window once again, click OK. So we have completed up to this. Next, analyze macromolecule, choose. Instead of choose, I told you to use open. Analyze macromolecule, open. Yes, that's it. The process is completed. Next, go for analyze conformations, load. Analyze conformations, load. That's it. We got the energy. So the negative, what we got for the post one is minus 5.62 kilocalorie per mole. This is the result, minus 5.62. But the second dog post is maximum minus 3.31. You know, if a ligand is binding well with a receptor, okay, we can tell the significant binding happens only if the dock energy is minus 6 kilocalorie and below. But our first dock post itself, we got minus 5.62, which is more than minus 6 kilocalorie. So whatever docking we have done is not significant. If docking is significant, we should get the docked energy as minus 6 and below. But we got more than that, even for the first post itself. So this docking is not significant. So Even I should show this, then only you can understand. Okay, so once it's docking I did, but docking has happened, but it's not significant. And what are the other output parameters you can get? Binding energy, other than binding energy, Ki, okay, which gives you the concentration, how much those we can fix for a drug. This can be determined from the Ki, and other kind of energy parameters are shown here. The next is to analyze conformation play rank by energy. Analyze conformations. Sorry. Analyze conformations play rank by energy. Now, after clicking this, you get a window like this. Click ampersand. After clicking ampersand, click build current. Now, write complex. If you click write complex, then only you can able to save your ligand and receptor. It is actually Indinavir, isn't it? Now I'm going to save a complex file as Indinavir protease complex. Now now we can save according to however we want to save. So the whatever file name I'm going to save, I save the file name .pdb in double quotes. Okay, if only if you save this complex .pdb in double quotes, then only it will be saved like PDB file. So after completing this step only, you have to close this windows, or else whatever you have done, all the files, you can't able to use it finally for analysis. So this is the step you have to use after completing the sixth step, then you can close it. If you see this demo 10, initially we had 5 files, but see how many files are created now. We had .map files, .fld files, .dlg, dpf, so many files are generated, isn't it? But our output file is Indinavir protease complex. Okay, for opening this ligand receptor, I'm going to open with BioVia Discovery Studio visualizer. I told you this is a freeware. Anyone can download this software in the BioVia website. So here, in this visualizer, you have to click View Hierarchy. If you click View Hierarchy, you can get information about the protein. If you see here, okay, if you want to get a 2D interaction, so here this is a 3D interaction of a ligand with a receptor, isn't it? So a ligand is shown here in the stick model, and the protein is in solid ribbon model. And if you want to see the 2D interaction, click Show 2D diagram. So the ligand interacting with binding site amino acids are shown here. If you see further, okay, in the 2D interaction, see, you have van der Waals interactions shown in light green. This is van der Waals interaction, glycine amino acid. Conventional hydrogen bond is there? Yes, is there. The oxygen atom is sharing with the threonine 8 conventional hydrogen. Pi sigma bond is here, isoleucine A54. Alkyl bond is there, pi alkyl bond is there. So this is a 2D interaction diagram of a ligand with a binding sites of a receptor. This is how we go for docking and analyzing the results. So initially I showed you how to go for finding the binding energy. Now I showed you how to find the binding site information. Okay. Any doubts you have, participants? If you have any doubts, you can ask me. Sir, at last, please tell why all the other exe files are required and what each does. So autograde 4.exe run is used for running the autograde. And autodoc 4.exe is used for running the autodoc. If you have these exe files in your folder, it is easier for you. It will be comfortable for you to find all the output files in a single folder. There won't be any confusions later. Okay, so this is the significance when cannot. And any other doubts you have, participants? If no doubts, thank you for your patience, and we can see the next session 6 on tomorrow. So thank you. Have a great day.